So should we play, which video should we play now? Wish.
Do you see potential for economic progress that also improves human and animal nutrition in countries with some of the fastest growing populations in the world? Do you want to partner to find solutions for the protein gaps found in Africa, Asia, and Central America? If you can join us in capturing business opportunities in nutritious foods and feeds, then stay tuned. Visionary U.S. soybean growers founded the World Initiative for Soy in Human Health, or WISH, as a program of the American Soybean Association. We are committed to strengthening agricultural market systems in developing and emerging economies in order to improve food security and build long-term demand for U.S. soy. Our work supports businesses, governments, and non-governmental organizations from Guatemala to Uganda to Cambodia to introduce nutritious foods and feeds that grow their businesses and economies. At WISH, we focus on regions where U.S. soy markets don't exist yet, blazing new trails in new sectors around the
YouTube
world.
and Facebook.
For example, WISH has made significant impacts in Central America by partnering with companies that use U.S. soy to manufacture more nutritious foods and beverages. In Asia, WISH partners have expanded their businesses by putting U.S. soy in fish feed for the first time. And in sub-Saharan Africa, local entrepreneurs have partnered with WISH to grow businesses that sell delicious and nutritious soy-based local foods, as well as feeds for poultry, fish, and more. So what can a partnership with WISH do for you? Working on behalf of U.S. soybean farmers, our goal is to create global partnerships and provide new opportunities for businesses and entrepreneurs that expand the potential for trade. Our approach is designed to increase demand, fuel economic growth, and connect entrepreneurs. The best part is that we do this at no cost to you. So how does it work? Thanks to decades of accumulated knowledge, we tap into expert resources to educate, mentor, and network to help your business grow both in size and through the latest innovations. Thanks to our soy experts, who are deeply connected to the regions where we work, we offer you face-to-face education and technical expertise. Working in developing and emerging markets, we identify market sectors with real potential for growth, work with key stakeholders, and provide effective trade solutions. While serving as an incubator, we connect driven leaders with supply chain partners. We help you network for strategic partnerships and create opportunities to launch a new product or service. And our go-to market strategists offer a localized approach, the right resources, the right expertise, and the right systems for better nutrition and improved food security in your market. At WISH, our greatest strength is our ability to facilitate strategic partnerships throughout the world to promote the benefits of soy. WISH and the American Soybean Association have the vision and the voice to continue to build awareness, create partnerships, and drive change for a healthier world.
Yeah. Hello, good morning.
Good morning.
Yeah. Right. So I see we have quite a number of participants who are logged in now. That's about 20 participants this morning. Right, thank you very much, and we'll try as much as possible to be on time. The beautiful part of it is that our presentations have been pre-recorded, and so barring any distractions, we should be able to proceed very much on time. Yeah, yesterday we heard a lot about the challenges and all the interest in aquaculture in Ghana. So we've heard of at least most of the stakeholders who spoke yesterday, those who are in position of authority, those who are in position of influence. Most of them have expressed their interest and their commitment to help strengthen the aquaculture sector in Ghana. Right, so we continue with the more academic aspect. But most of our presentations will have direct applications and implications in strengthening and solving the challenges that have been mentioned yesterday. And so without much ado, I would like us to listen to the various presentations. And please, as you listen, you jot down all your questions that you have after each presentation. We have the opportunity for the question and answer sessions. And so each question and answer session will just be about approximately 5-10 minutes, depending on the interest and the questions that are available. But we'll try as much as possible to be on time. So thank you very much, and we'll start with the first video, which is will be played to you. Thank you.
Good
Yeah,
morning.
so
My name is
please,
co-host.
co-host,
Presenting on the topic investigating pathological agents associated.
Can you please play the video?
I hope you can see my screen for the video.
Yeah, I see it very well. So please carry on.
Morning. My name is Recirculation. I'm presenting on the topic investigating pathological agents associated with the rusty brown spot on the Nile tilapia, Recirculation niloticus, cultured in Ghana. My co-authors are Dr. Samuel Dodu, Dr. Michael Wiafikwajan, and Dr. Samuel Ado. The outline includes an introduction, objectives, the justification for the project, materials and methods, results, discussion, some recommendations, and references. Now, in the wake of reduced fish population in our oceans around the world has turned its attention to aquaculture to be the backbone of fish production all around. Ghana is included in the drive to improve and promote aquaculture production. However, disease has become a major constraint in the growth of the aquaculture industry around the world. Diseases can be categorized as infectious diseases, where infectious diseases are those that are caused by pathogens such as viruses, bacteria, and non-infectious diseases are those that are directly related to environmental conditions such as change in temperature, water quality, among others. In Ghana, it has been found that the most predominant fish pathogen that is causing most of the problems on our fish farms are the Streptococcus and Flebobacterium, that is for bacteria, and then Aclea saprolegnia for fungi. Now, many fish infections have been known to cause mass mortalities. However, it is not every one of the infections that is capable of causing mass mortality. Some of them do not cause mortalities, as it were, but reduces the presence or the appearance of the fish, or just presents a chronic condition. Some of them make the fish unsightly. Some of them prevent the fish from growing as it should. Now, these are examples of the fish that were collected for this the purpose of this research, and that is the rusty brown spot, as can be seen on the skin of the tilapia. Now, the objective of this project was to provide information on the pathogens, possible pathogens that are associated with the rusty brown spot on organism, or the Nile tilapia, as has been reported in Ghana. Again, we wanted to dive into the microscopy and biochemical analysis to determine exactly which pathogens are related to the rusty brown spot as best as possible. And then also to determine whether these pathogens are present in the sediments or in the water or in the feed at the Aquaculture Development Center. Now, the idea, the justification for this research was to ensure that Ghana is not going to spend so much money to treat diseases that were directly associated by the consumption of these fishes. Some of the diseases that have been known to be transferred from fish to humans include gastroenteritis, bacillary dysentery, typhoid, cholera, among others in Africa. Again, we wanted to be sure that we can get to the bottom of the cause of this disease, because we do not want this consumption to affect the quality of fish that is consumed in Ghana. So this is the study area, that's the Aquaculture Development Center, where the arrow is pointed is the pond from which the samples of fish were taken. Now, the materials and methods used were two. We used two processes to determine these organisms. We did mycological study and the biochemical reactions to determine fungi and bacteria possibly present. Now, for us to be able to work in the lab with the fishes being fresh as possible, we conveyed them in plastic bags filled with oxygen alive to the labs. And for the water, feed, and sediments, we collected them in sterile bottles and Ziploc plastic bags to the lab. With the mycological study, what we used were three different media. We used a potatoes dextrose agar, Tepadox media, and then Richards medium. We also took samples of fish skin, cut them up, and plated them directly on all the three media. And then for the feed, water, and sediment, we did serial dilution up to -3 and inoculated them on the plate. Then we allowed the plates to incubate for 5 days at a temperature of 28 degrees Celsius. And after the 5 days, the fungal growth was stained with lactophenol blue and viewed under a microscope, and the morphology of the mycelium and conidia were used to identify them based on the Barnett and Hunter 1976. For bacteriology, the skin from the infected fish were collected and homogenized, and the homogenates were extracted and spread over 2 agar. We used the tryptic soy agar and the tryptone yeast extract salt agar. The plates were incubated at room temperature for 24 hours, and the growth colonies were observed using their morphology. Then afterwards, pure cultures were made, and then we used the full identification scheme, such as gram staining, and biochemical activities to determine which organisms we were dealing with. So these are the results that we had. Looking at this graph, we see that from the isolate of fish, water, and sediment, that is the infected fish, the water, the sediment, and the feed, we identified that Aspergillus was the genera of fungi that was predominant, followed by Penicillium, and then Mucormycete, Rhizopus, Echinacea, Fusarium. Those that were in least present were the yeast, Trichoderma, Syncephalastrum, and then Cladospora. They were all present in different numbers of fish, but they were present in the feed, the water, and the sediments. Again, when we take a look at just the rusty brown spot, Aspergillus comes through too and was present on all the fish samples that were taken. 30 fishes were used, and all the fish samples had Aspergillus on them. And then it also had some of them, about 5 or 6 of them had Penicillium trichoderma, and just about 3 or 4 fishes were found to contain mucor, yeast, and then Echinacea. Now, when we take a look at this table, the Aspergillus species that were identified from the fish, the water, the sediment, and the feed, these species were present in all the samples that we worked on, were the Aspergillus flavus and then the Aspergillus niger. They were present in every sample that was worked on. The bacteria cultures that we did of the skin sample yielded 6 isolates of bacteria, and then the biochemical test that we used went further to indicate the possible presence of Flavobacterium, Pseudomonas, Aeromonas, Streptococcus, and then Francisella. So these are the biological or biochemical reactions that were used to identify the isolates. We used the motility test, gram staining, oxidase test, catalase test, TSI, and then SC to determine which organisms we were dealing with. But after that, we did a confirmatory, went further to do a confirmatory test using the Brucamaldi biotyper to further confirm the presence of specific pathogens. And these were the pathogens that were isolated. We had the Pseudomonas aeruginosa, Klebsiella pneumoniae, Streptococcus pyonis, Streptococcus aureus, and Flavobacteria species that we could not identify which species it was. So, in looking at the results that we've had, we come to an understanding that fish infections are as a result of not just one particular organism, but an interaction between bacteria, oomycetes, and other fungi that are present in the environment. We also come to an understanding that multiple infections of bacteria and fungi increases the severity of disease. Once an organism is infected with a bacteria or a fungi, the severity increases because of the presence of either of them in the environment. And we also have come to an understanding that fish infections are caused by some species of fungi from the genus Penicillium and Aspergillus, as stated by Yacoub and Gozlan, respectively. From our results, we see that Aspergillus was in high incidence or was present in virtually all the samples. However, we know that Aspergillus is ubiquitous in the environment, and fishes are very susceptible to Aspergillus infections. Aspergillus flavus and Aspergillus niger, including fumigatus, have been known to cause high effects or to have very great effects on fish health. And Saprolegnia and Eclair have also been documented to cause infections in fish cultures. Now, all the bacteria that were isolated have been known and documented to be pathogenic. Bacteria such as the Streptococcus agalactiae is known to cause mass mortality in fish. However, Saprolegnia innei reduces the immunity of fish, making it susceptible to other diseases. Now, in conclusion, we now understand that the rusty brown spot cannot be attributed to just one pathogen. Can be associated with an ensemble of pathogens. Some of the mycological and bacterial pathogens that we isolated as known to be zoonotic, and some of the pathogens form part of the fish's fauna already, or the environment of the fish, so cannot be categorically stated that it is the cause of the disease. However, we would want to recommend that a challenge work should be done to ascertain the exact cause of the infection, and also to know, to make sure that feed samples from major feed producers are tested and taken through these processes to ensure that the pathogens are not being transmitted from the factories to the fish ponds. And then also we have to create awareness with the farmers to engage in best management practices on their farms. Thank you very much and have a good morning.
I've just muted.
Yeah, thank you very much to our virtual presenter, Denu. Yeah, so this was a wonderful presentation about the rusty brown spots. Yeah, we'll take the opportunity. I hope the presenter is online now. The presenter or any member of the team.
Yes, the presenter is here.
Great, wonderful. Right, so we ask questions, we invite questions from our audience. Now we have about 50 participants. And whilst we are waiting for the audience, our participants to ask questions, I would like to take the initiative to ask the first question about the prevalence of this rusty brown spot disease. how prevalent is it in Ghana or in the industry, and especially your study area, ADEC?
Okay, thank you very much for the question. So, we received reports from farms in the Ashanti region, and then ARDEC, among a few other farms that reported of the prevalence of that rusty brown spot on the tilapia. Initially, the farmers attributed it to probably change in temperature or other environmental conditions, but they realized that season in, season out, as they restocked their ponds, these brown spots re-emerged after a period. So it became a source of worry for them, because then what it means is that when they send it to the markets, nobody would want to patronize their fish because of the way the fish. So yes, it is found in a lot of ponds. However, because it doesn't kill the fishes immediately, many farmers, I'm sure, fail to report it because it doesn't appear that it's a major challenge for them. But I think that if we don't get to the bottom of the cause of it, maybe eventually it might become a bigger problem for the farms. So yeah, it's quite prevalent on farms.
Right. So we ask Bulahami, please unmute yourself and ask your question. Bulahami, please unmute yourself and ask your question.
I've told him, so he has made the first person. They've raised their hands.
Right, and then maybe we go to Nuruddin, please.
Sorry?
Nuruddin, unmute yourself and ask your question.
Thank you very much.
Okay. Okay.
It's good to be here, and also I want to say a very big thank you to the presenter. It was a very short, precise, and very educative presentation.
Nuruddin, we can't hear you.
Oh, sorry. Can you hear me now?
Yes, please.
Okay. Sorry, I was saying that I'm very impressed to see the presentation, because it's rightly addressed some of the recent occurrences that have happened in tilapia farming in Ghana and also in the Republic of Benin. And I know it has been some of the hindrances to the success of some of the tilapia farming right here in West Africa. So I want to quickly ask some questions and also make some points on the recommendation of the presenter. And 3 recommendations were made that further findings should be made on the causes of the disease. And secondly, it was mentioned that feed samples should be taken. Then also that we need to check our farm practices. So I think basically I want to challenge ourselves here and also the farmers that I want to start from the last, which is the best farming practices. You agree with me that some of the issues that we've been having or challenges we've been having here in Nigeria, in some other farming, especially catfish to export, is because of our farming practices. And I know that it is time in Africa that we need to take it, do it right. We need to do it right and make sure that hello, there's an so we need to start doing it right and pay attention to
Nuruddin, can you be brief for the sake of time?
Oh, okay, sir. So I will quickly submit that we need to pay more attention to the farm practices. Very essential that we need to do more awareness and sensitization of our farmers, that when we're establishing farm, don't let us do our farming as soon as we are doing peasant farming. Farming should be a business. We should have all the quality assurance officers in place to follow the. I think it is important too that here in Africa we need to make sure that we pay attention to the feed. We should not be looking for the cheapest available feed, because it's going to be detrimental to our operations. So I will challenge every one of us to please see how we can do more sensitization on our farmers, so that we can take this into the next commercial level by paying attention to the feed we use, and also to be very, very prudent.
Thank you, Nuruddin. We have Nuruddin. Thank you. We have other people. For the sake of time, let's be brief. Let's go to our next person, Ogbe. Please, my right, your hand, please.
Good morning.
Sorry, I couldn't mention your name.
No problem. No worries at all. I just want to appreciate the presenter once again, but I just have one question. Now, I want to know what influenced the presenter's choice on the number of ponds they use. During the presentation, she mentioned that they just from one particular pond. So I want to know what informed the choice of choosing one pond, because the issue of doing more research as to ascertain the number or the causes, the etiology of the disease would have been a bit addressed if they are taking samples from a wider population. So I want to know why the choice of one pond. Thank you.
Thank you very much, madam. All right, so at the time of the research, based on the location of the school and the research center, it was more prudent to work directly with them because one, they are the source of fingerling production to many of the farms in Ghana. And so we thought that if we could find out the causes, we could work on that farm, we could probably prevent occurrence on other farms. But then at the time of taking the samples, the other ponds had been emptied of tilapia. What they had was smaller sizes. And the irony of the disease is that when they are small, like the juvenile stage, the disease is not present. It is when they gain weight to about over 20 grams, 30 grams, 40 grams, when they start maturing, that you actually see the disease present. So that is why we didn't just work on the samples, the fish samples themselves. We took a look at the feed, we took a look at the water quality or the water sample, and then the sediment, with a knowledge that if they are young, probably the disease would be the body of the fish might act as a host, but as they grow and interact more with the environment and other conditions in the water, then it allows the disease or the condition to become expressed. So basically, it was, and that is why my recommendation indicated that there is a need for a challenge experiment. There is a need for us to go back to that environment and other farms to be able to, work on this organism. We need funding, a lot of it, for that process to happen so that we can actually cover the entire nation, to be sure of it. But yes, so the first thing we need to understand is that the condition is not present on juveniles. It only starts showing when they start maturing. I hope I've answered the question adequately, madam.
Yeah, thank you very much. So, thank you. I think we are 3 minutes behind time. So we go to our next presentation, and we are very grateful. And anyone who has questions, please, there's the chat box. Bola, we are very sorry. We have to move on to the next presentation because we are short of time. You could just send your question, put it in the chat box, either in private to her or general. We can all contribute.
The next presentation.
Good morning, fellow participants here seated. This is my name. I'll be presenting my research findings on prevalence of Heming parasites in pond-raised Phallus grapinus in Odela local government area of Ogun State here in Nigeria. As a way to begin, the aquaculture has been rated as the fastest growing food production subsector in the world and in Nigeria, to be specific. There has been a tremendous increase in the aquaculture industry, especially fish farming in Nigeria in the recent past. However, with this growth and intensification comes the problem of diseases and parasites. Parasites and diseases have been a sizable cause of loss to the aquaculture industry, specifically the fish farming subsector, globally and in Nigeria. Now, this cost of loss can either be directly or indirectly. Directly, either through production losses, through increased operational costs, or indirectly through costs to society, such as reduced availability of the products, loss of income, loss of employment, and much. What is the problem and why this study? Now, number one, human population is on the rise. So is a corresponding demand for fish and fish products, so as to be able to meet the ever increasing demand for animal proteins recommended by standard bodies. This is seen in table 1. Number 2, there have been fluctuations in capture fisheries. In fact, of recent, there are reports that capture fisheries has reached its peak and is already declining. And there have been increased focus on culture fish. This has resulted to associated effect of parasites and diseases. Number 3, there's also a wide gap between demand and supply of fish products. Table 1 shows the world fisheries and aquaculture production and utilization in million tons between 2011 and 2018. Now, in 2012, there was a decline from 92.2 million tons recorded in 2011 to 89.5 million tons in 2012. Also, in 2016, there was also a decline from 92.7 in total capture production in 2015 to 90.9 in 2016. However, total production from aquaculture has continually been on the rise from 2011 to 2018. Now, similar findings or similar report is also seen for utilization of the product and also population growth. Now, there has been a continuous increase in utilization of the product and also growth in human population. Now, this table is sourced from FAO 2020 and UNDESA 2019. Now, for demand gap for fish in Nigeria, as of 2014, the production of fish product represented by blue stood at 1.12 million metric tons, while demand stood at 3.32 million metric tons. You can see the wide difference between demand and supply of fish. What then did we do? Now, let me just call your attention again to the fact that the scope of this study is to investigate the prevalence of parasites in cultured fish from the study area. Now, in the study area, we sampled or we purchased 60 fish samples from 3 different zones in this area. Now, from each of the zones, 2 farms were investigated. The study area covered a period of 4 months. Now, purchase samples were taken to the parasitology laboratory of the Department of Microbiology and Parasitology of the College of Veterinary Medicine, Federal University of Agriculture, Abeokuta, for parasite identification. Sample preparation. At the laboratory, live fish were immobilized using 4% chloroform. The length and weight of the fish samples were taken using measuring board and sensitive scale to the nearest 0.1 centimeter and 0.1 gram respectively. Fish were then grouped into different class groups, as will be seen in table 2. Sex identification. Sex identification was done by visual observation of the samples. Laboratory procedure for parasite isolation. Fish samples were dissected in the midline so as to expose their visceral organs, with the body wall deflated to have a better exposure of the viscera. The gastrointestinal tracts were removed from each of the samples and cut into the stomach, esophagus, and intestine, and these same specimens were placed in separate petri dishes containing saline solution. The specimen, that is the stomach, esophagus, and intestine, were dissected and viewed under the microscope. Parasites isolated were preserved in 5% formalin. Identification of Hemings. Identification of the parasite was done using the Atlas of Parasitology of CDC DPDX. Laboratory identification of parasitic diseases of public health concern as reported by the Prevention Center for Disease Control 2015. Table 2 shows the length and weight distribution of the samples during the study. Analysis. Prevalence was analyzed according to Bush et al. 1997. That is, number of infested hosts by one or more parasites divided by the number of hosts examined for that species. Relationships between factors such as host sex, weight, length, and parasitic infection were obtained from data using chi-square. Data collected were analyzed using the statistical package for social sciences, version 20, and Microsoft Excel 2016 for Windows. Results and discussion. Plate 1 shows the different species of any parasite isolated from the sampled fish. 3 any parasite belonging to 2 classes of parasites, nematodes and cestodes, were isolated from the sampled fish. Plate A shows Diphyllobotrium latum, Plate B Dipylidium caninum, and Plate C Necator americanus. Now, the presence of Dipylidium caninum in the sampled fish cannot be well explained, as this parasite is known as dog and cat parasite. However, it could be due to the overgrown vegetation around the pond, which could have resulted in the carrier fleas or intermediate vertebrates to lay their eggs around the fish pond, which may have gotten access into the fish environment by chance. And according to Bowman 2014, the wild pathogenicity of this cat and dog parasite can result to weight loss and anorexia. 3 shows the percentage prevalence of the intestinal hemings of Claras garapinos. Now, from the table, we observe that Diphyllobotrium latum had the highest number of infected fish and the highest prevalence, standing at 6.67%, as opposed to Dipylidium caninum and Necator americanus with percentage prevalence of 1.67. The high infestation of this parasite, like I said before, could be due to the overgrown vegetation and the presence of crustacean copepods, which were observed around the fish farm. These are the intermediate carrier of this parasites in the environment. Now, according to Ede and Solomon 2016, Diphyllobotrium latum, commonly known as a fish tapeworm, is the biggest tapeworm in humans and causes the disease Diphyllobotriasis. Similar result was reported by Biu and Akorede 2013. 4 shows the prevalence of the prevalent percentage of heming parasite in relation to sex of fish. From the table of the 60 fish examined, 35 were males and 25 were females. However, of the 35 male examined, only 1 was infected, giving a percentage prevalence of 2.86, as opposed to 20% prevalence observed for the female fish. However, there was no statistical difference. In general, the low prevalence of parasites recorded in this study could be due to good water quality monitoring and management carried out by the farm owners. Similar result was also recorded in the study of Ibironke and Morenikeji 2018 from these same species, although from different water bodies. This was, however, not the case in the study of Adiogo et al. 2014. Again, relating to sex, the high prevalence in female could be attributed to the physiological state of the female and the different degrees of resistance put up by the different sexes to parasitic infection, infestation as reported by Emery and Egbe 2006. This again was not the case with the study of Kawe et al. 2016. Figure 1 shows the prevalence of hemings in relation to length. Shows the prevalence in relation to width. Now, from the figure 1 and 2, it is seen that the prevalence of this parasite was higher in the larger size fish than for the smaller size fish, although there was no statistical variation at 95% confidence limit. The high prevalence with increase in size of the species could be attributed to the distance that the bigger fish will have to travel in search of food, and also due to the buildup of parasite larvae in the fish as it ages. This finding is in line with that of Omeji 2013 and Tasawa 2007, but did not agree with the research of Biu and Akorede 2013. In conclusion, the study revealed low infestation and prevalence of heming parasites in pond-raised Claras garapinos from the study area. Also, Diphyllobotrium latum, Dipylidium caninum, and Necator americanus are the heming parasites observed in this study. Thirdly, we also report that for the first time in this study area, the parasite Dipylidium caninum and Necator americanus are being observed or isolated from pond-raised Claras garapinos. These are my selected references, and thank you for listening.
Thank you very much. We are very grateful, Ogbenechukwu. Now we invite questions once again. Let's be brief, no lengthy introductory comments. Direct comments, please. We invite questions from our participants, please. And whilst you wait, Ogbenechukwu, you said the study reveals a low prevalence of helminths in fish. Do we assume that a prevalence rate of about 6 is low?
Oh, she just dropped.
Ogbenechukwu or any other members of the team, are you available to answer questions?
You can say she was, but she just having connection issue, I presume. Or should I say it?
Follow. Okay, it looks like yet. So maybe later on when we end, we just check up again and see if any of them have
here. Can I ask my question for Luke?
No, you are there, but the team members are not available to answer the questions.
Oh, I see.
They were available, but I think their connection just dropped just now. So I presume they would be unable to respond. We're trying to
time
see.
At the end, we ask them, right. At the end, we'll ask if they are available to answer the question. So, Foloke and Nuruddin, please, let's hold on with our questions later.
Thank you.
Right. And so, please, let's continue with the next presentation. That's on induced breeding.
Good day, ladies and gentlemen. I am Kennedy Senanu Ameti, presenting this paper titled Induced Breeding of the African Catfish Under Varying Broodstock Ratios Using Overprint. This paper was co-authored with Dr. Samuel Addo of the University of Ghana, Marine and Fisheries Science Department, and Dr. Winnie Sowa of the University of Ghana, Marine and Fisheries Science Department. The order of my presentation is as follows. The African catfish is a freshwater fish that forms part of many traditional meals in Ghana. The commercial production of this fish is, however, limited by many challenges. Chief among them is that seed for commercial production is not readily available to farmers due to the challenges of breeding the fish in captivity. However, most hatcheries resort to artificial methods of propagation, where usually the male breeder is sacrificed in order to produce seed. The objective of the study was to induce breeding in the African catfish under varying broodstock ratios using overprint. Specifically, we sought to induce spawning with overprint, assess the breeding success, and then assess larval production between the artificial propagation method and the semi-natural propagation method. The study was conducted at Flowcell Farms Limited, located in the Volta region of Ghana. Healthy broodstocks were procured and preconditioned for a month. The A complete randomized design was used, and brood stocks were divided into 3 spawning groups in a ratio of 1:1 male and female for the artificial spawning group, 1:1 male and female for the semi-natural spawning group, and also 2 females to 1 male in a semi-natural spawning group. All females were injected with Overprim at 5% of their body weight, as shown in plate 1. Stripping and fertilization. For the artificial
when you're talking in Ghana, it's
propagation method, eggs from females were stripped into bowls and weighed. The males in the same group were killed and the gonads were removed to extract the sperm to fertilize the eggs. The fertilized eggs were incubated on incubated trays as shown in plate 3.
Is it normal voice call, the way you made this call, that it was so clear? Over there,
females and males in the semi-natural propagation group were kept together in spawning tanks, as shown in plate 3, after the females had been injected with Overprim. Estimation of breeding characteristics were done as followed: relative fecundity, fertilization rate, hatching rate, and survival rate. Water quality was monitored during the breeding process. pH, dissolved oxygen, and temperature readings were all taken. All data collected were analyzed for analysis of variance run on SPSS. The results in table 1 shows water quality parameters that were recorded during the experiment. The values for temperature, dissolved oxygen, and pH did not differ significantly among the experimental groups. Spawning was successful across all experimental groups as indicated by table 2. Differences between the artificial and semi-natural spawning groups was as a result of partial ovulation as reported by de Graaf and Jehans in 1996 in research. Differences observed in fertilization could be due to stress induced by the aggressive behavior of brooders in the semi-natural group. This led to injuries as shown in plate 4. It's a picture to depict the injuries that occurred among brooders that led to some mortalities, especially in the semi-natural spawning experimental groups. Table 3 indicates that there were no significant differences in hatchability across experimental groups due to the stable water temperature shown in Table 1 previously. Differences in larval production was due to differences in total eggs produced, in that there were more eggs produced in the artificial spawning groups compared with the natural, semi-natural spawning groups. In conclusion, ovulation and spawning success without sacrificing the male broodstock treated under semi-natural reproduction is possible. Also, broodstock ratios 2 females to 1 male affected the quantity of seed produced. Artificial reproduction technique produces more catfish seed compared with the semi-natural technique. It is therefore recommended that semi-natural reproduction technique could be adopted by small hatcheries as a means of producing catfish seed. I would like to acknowledge the following for their support during the carrying out of this experiment. Thank you very much for your audience.
Looks like we've lost the moderator. Yes. Do you want to take charge? Kennedy is ready to answer questions.
Hello.
Okay, he's back.
Who is asking? I guess my thing temporarily went offline. So, for Luke, your question, please.
Okay, thank you very much. I want to thank the first presenter for a beautiful presentation, but there were things that came up to my mind on the presentation and which calls for urgent attention. The strange parasite that she said could be due to cat or whatever is an issue that shouldn't even have occurred if there had been good sanitary measures in place. And that calls to question the fact that most people practice and do not belong either to associations or are not in touch with supervising authorities are likely to have those issues. Because if they have been in touch with federal department officials for the inspection of the farm, they are not likely to have that. And has she thought of the possibility of the lies coming from human beings? I'm just suggesting that. And the second presenter, thank you for your presentation. And I have an issue with your very first quotation of 2004. It is a bit too late about being difficult for catfish to breed in captivity. I think Nigeria has achieved a lot in that area. And with that issue does not play out again. Thank you very much.
Okay, yes. So
Nuruddin, please, we are listening to you.
Thank you very much. Not to waste time, I think Dr. Areola have actually hit the point on the first topic. And but my concern about the first topic is for us to pay more attention to the transmissibility of this emit to the consumer. So we need to do further study on to monitor whether this emit is being transmitted to human being during consumption, because we know that most of our catfish, the cooking is always mild cooking, so that we can get all the protein contained in the fish. So we need to do more study on this. Then quickly to the last topic that is being done. You know, doctor have said it. I think we need to get ourselves updated in Africa, especially West Africa, because Nigeria, we've gone so far on this spawning process in terms of using induced or using the natural pituitary gland of the male. So we need to we need to find a way to synchronize our data and also collaborate, because some of the presentation is outdated. And secondly, why the fish, the injected female got injured? I think there must be something wrong in the compartment where you put them. Of course, definitely they will be very aggressive, but there are ways that we're doing it now that you will have reduced those injuries. And it's very sad to be seeing mortality and injuries like that during spawning. So that's just my observation. We need to synchronize and collaborate to see how we can assist ourselves, because Nigeria is far ahead in terms of catfish spawning and using induced or the natural way. Thank you.
Right. Thank you, Nuruddin. I think that's the purpose of this conference. We are getting to know each other, getting details, and I think behind the scene, we'll be contacting each and every person and trying to draw these collaborations. Godfred, can you please go on and please be brief? Godfred Tete, unmute yourself and take your mic.
Yeah, thank you very much for the chance to ask a few questions. I want to ask the presenter on the inducement of catfish. What informed his decisions or his recommendations that smaller farms should rather practice natural spawning as compared to the artificial? What exactly is the reason behind? Is it because of the cost of overtrain, or what? We want to know.
Yeah, please, we are listening.
Hello?
Yeah.
Can you hear me?
Yeah, I can hear you, yeah.
Yeah. Well, what informed that decision is because, you see, in Ghana, we don't have very large established hatcheries that are able to produce enough seed for farmers. And catfish, unlike Nigeria, in Ghana, is produced on fairly small scale. And since some amount of seed can be realized from the semi-natural technique, it will be prudent to use that, since that is fairly less involving compared to the artificial and other means of producing catfish. That's what informed the decision to recommend that technique to small farms.
Okay, right.
Thank you very much. Orisana, please unmute yourself and let's hear your question.
All right, thank you very much. I have some major concerns with the second presentation. And the reason for the concern is not far-fetched. The justification for the work, I think, is not made very clear. And I know the whole essence will have been to ensure, to prevent the sacrificing of male fish for artificial propagation. However, in your result, there was a plate where you showed a number of mortalities in the paired catfish, meaning that the justification is nullified. Because there's no, in most cases, as a character of the Clarias garipinos, they become aggressive when they are paired. You understand? So the justification for this work, I think, should be made clearer. Another thing, I saw a table where means were separated. I don't know if there are replications. I only saw the pairing for the artificial 1-to-1. There is the semi-natural 1-to-1 and the second 1-to-2. Are there replications for each of these treatments? If there are no replications, then I don't think it will have been possible to separate any means. Those are my concerns. Thank you.
Right. So,
Okay.
Kennedy,
Yeah.
please, can you respond to the second part? The first
Yes,
one is
yes. I think the comments are noted. Then on the replication part, yes, there were replicates for each of the groups. They were replicated. So you could see that you have even standard deviations on the mean. So there were replicates for each of the spawning groups.
Right. Thank you. Now, our last comment from Bola Hami.
Yeah, thank you very much. My questions goes like this. You said that you were able to observe fertilization rate and hatching rate. How were you able to do that? Did you count all the eggs that were produced and then find the percentage of those that are, or you had a small portion somewhere within the same system that you did that? You didn't explain to us how you achieved it. Did you hear me? Was my question clear?
I don't know if the technological challenge was from here. I didn't hear your comment that much. But please, Kennedy, if you heard him, please respond to it.
I didn't get the comment. If you could write them in the comment chat or the chat, then I could probably respond to.
Yeah, Bola Hami, your net was pretty bad and we couldn't hear the comment. So please send it as a chat and we'll respond to that appropriately. Right. So for the sake of time, let's move on to our next presentation. Yeah, that will be on age and group of reticulate fish. Hey, no, sorry. That's evaluation of tilapia. That's bilaterals.
Good morning to my audience. I'm Joyce Buenokinluchot. The title of my work is Evaluation of Nile Tilapia Fingerlings Production at the Aquaculture Demonstration Center, Ashama, Ghana. The outline is introduction and literature review. We talk about materials and methods. We will discuss the results, conclusions, and a few recommendations. Let's look at the introduction and a bit of literature review. In 2015, aquaculture supplied 106 million metric tons in live weight, which was nearly half of the total production of fish globally. Aquaculture appears to have the potential to make significant contribution to the increasing global demand of fish. The Nile tilapia is widely produced because of its quick reproduction rates, is able to tolerate hard environments, is able to endure disease conditions, and it can be cultured under numerous and diverse farming systems. In 2015, the fish seed deficit in Ghana was 50 million on the average. The inadequate supply of tilapia seed, mostly fingerlings, is considered one of the major constraints to the development of aquaculture. Why the center was chosen? The Aquaculture Demonstration Center, which was set up in 1965, in 2016, produced 1.2 million tilapia fingerlings. This level of production could probably increase if scientific research is carried out to evaluate the hatchery activities at the center. The aim of the work. was to evaluate the production characteristics of Nile tilapia fingerlings at the center for improved productivity. 3 objectives were considered. The first one was to evaluate the spawning characteristics of the brooders used at the center. The second was to determine the growth performance of the fry and fingerlings under their culture conditions. The third was to assess water quality in the culture facilities used in fingerling production at the center. This is an example of a culture facility at the center, a 50 square meter tank, concrete tank that was used for the work. Now, for the brooders, the females were of an average size of 150 grams. The males weighed 218 grams on the average. Now, these brooders were stocked at a ratio of 3 females to 1 male, at a stocking rate of 4 brooders per square meter. Each tank was stocked with 200 brooders, 150 females and 50 males. These brooders were fed ad libitum for 12 days before fry was finally harvested. These are pictures of fry harvesting and brooder selection. The first one was surface harvesting of fry. The second one, total harvesting of fry and brooders. Now the fry harvested were stocked, they were estimated, weighed and stocked into 1 concrete tank. The brooders were, eggs were collected from the brooders. They were separated, rested, and then restocked again. Let's talk about the hormonal treatment of the fry. Fry harvested were stocked into 50-meter square concrete tank for hormonal sex reversal. 17 alpha methyl testosterone was the hormone used for sex reversal. This hormone was incorporated into 250 ml alcohol of 95% concentration. It was mixed into 1 kilo feed of particle size 0.1 to 0.3 mm. This feed was shade dried after thorough mixing. The fish or the fry were fed for 28 days on this hormonal feed that was prepared. This picture shows the mixing of the hormonal feed and shade drying of it. Now let's come to fingerling production. After the hormonal treatment, fry were harvested, counted, weighed and transferred into an earthen pond under minimal stress. This was to grow the fry to fingerling stage. In the nursery pond, the fish were fed 4 times a day with a complete feed for the period of 24 days to satiation. The particle size was 0.6 to 1.2 millimeters of 48% crude protein. Now, after 24 days of growth in the nursery pond, the fingerlings were harvested. The total number and average weight of fingerlings were estimated and percentage survival calculated. Fingerlings harvested were then conditioned for packaging and transported to outgrowers' farm. These were the results. The mean water quality parameters that were taken were morning and evening temperatures range between 27 to 31 degrees Celsius for the brooders, fry, and fingerlings. The dissolved oxygen rates were between 3.5 to 4.03 milligrams per liter for the morning and evening. pH levels were between 7.3 to 7.5. Salinity levels between the fry, fingerlings, and brooders were 0.23 to 0.29 on the average. Ammonia levels were between 0.01 to 0.12 milligrams per liter. Variables for the brooders used, as said earlier, 150 grams female size, 218 gram male size. Total number of females used in the 9 tanks were 1,350. The average number of eggs per female was estimated at 500. Estimated total number of eggs from all the females used was 675,000, and these eggs were hatched in the lab. Estimated total number of fry harvested was 138,631. Percentage hatchability from the 675,000 eggs was 20.5. So at the breeding stage, there was actually very low hatchability. That was stated 20.5, as said earlier. At the fry production stage, the final average weight observed was 0.23 grams. Survival rate was 79.5%. The feed conversion ratio was 1.46. Specific growth rate was 6.5 per day. The final standing crop was 207,575 kilos per hectare. At the fingerling stage, final average weight was 2.47 grams. Survival rate was 75.3%. The feed conversion ratio was 1.09. Specific growth rate was 3.26 per day. Final standing crop was 2,034,790 kilos per hectare. There were some few recommendations. I further recommend that further studies should be carried out at causes of low hatchability at the center. Then artificial and then natural hatching of the eggs should be done. Certified institutions should be put in place for brooder supply. And then breeding programs should be outlined to inform hatcheries as to how to operate. These are my reference acknowledgements. Thank you.
Yeah, thank you very much, Lutrot. Yeah, so now we invite questions from our colleagues. Yeah, please, questions, please. Lutrot, are you online?
Yes, I'm online, sir.
Great. Okay. So please, Orisana, question. Your question. Unmute yourself and ask your question.
Okay. The name is actually Orishashono.
Orishashono. Thank you, yes. Orishashono.
Yeah. Now, thank you very much for the presentation. I have one question for you. The culture system in place in the center that you studied, where you carried out your evaluation, I didn't really get that, because I think that will have an influence on their production, on the level of production. And again, that I think will help you to recommend, because the whole idea of this study from the beginning that I got is on how to improve production in that center. So if you do not have an idea of what the culture system is like, the size, are they putting the facility available to optimal use, you might not be able to recommend, because you said in the course of your presentation again that hatchability rate was around 20.5, which I think is rather low. It's too low. And because of that result, what recommendations do you have for the center to improve on their hatchability and ultimately the production of tilapia? Thank you.
Okay, so the eggs will actually hatch at the laboratory.
Joyce, please, we cannot hear you. You seem to be far away from your mic.
Hello, please can you hear me?
Yeah, it's a bit better.
Okay. So the eggs were actually hatched at the lab.
Wow.
Out of the 600
It's still pretty bad, please.
Hello, Doc. Hello. Hello.
Yeah. Okay, you go on. Go on.
Okay. So the system that was used for, we used the constant and then we used the constant. The boulders were actually brought in the concrete tank, which was the 50-meter square concrete tank. And then the fry treatment, which was the process diverter treatment, was also done in the concrete. But the hatching of the eggs were done in the lab. And out of the 600, that was how we got the 20% hatchability. So I was recommending that further work be done on the fry collection, which would be done in 14 days, and then eggs also should be hatched so that there is a comparative comparison between the two, and then we find out what exactly the problem is with the Hello.
Thank you, Joyce. I don't know if it's just from my end, but it's pretty difficult hearing you. Yeah, please just continue, put your comments and your responses through the chat. I think that might be the best way to overcome this technological challenge. And then we move on to our next presentation for the sake of time. That's Oluwale on age and growth of reticulate night fish.
All protocols duly observed. Welcome to my presentation on age and growth of Papiconum alpha, an emerging ornamental fish in Nigeria. This is the outline for my presentation. The subject matter is fish. Fish is a highly nutritious food, and a large-scale local production of it at cheaper prices will benefit the health of many of our people. Also of paramount importance is the fact that fish make ideal pets in an ever busy and urbanized society. Unlike dogs, cats, and even birds, they are quiet. They don't need to be walked or exercised. They don't give people allergies. They don't get lonely if you don't give them a lot of attention, and they take up relatively little space. Papiconum alpha is becoming important in Nigeria fisheries because the juveniles are ornamental. The caudal fin is in conflict with the anal fin, giving the outline of a knife edge, and hence the name knife fish. This fishing is based on white collection, and white collection is inherent with challenges including inadequacy, seasonal availability, and heavy post-harvest loss. Natural population of fishes are depleted to meet the increasing demand of the international and local ornamental fish trade. The number of collectors have increased, and there will be a guided diversification to wide range of species. Decline in abundance of Papiconum alpha are driven by increasing demand in ornamental fisheries. Several reports indicated that indigenous ornamental fish species in developing countries are depleted due to their foreign exchange potentials. Estimated global value of ornamental fishery is 15 billion per annum, and export of ornamental fisheries has increased. In spite of the socioeconomic importance of ornamental fisheries, including foreign exchange earning, employment, and recreation, little attention is paid to the declining populations in Nigeria waters. Available information of Papiconum alpha is fragmented, despite being harvested for human consumption and for the aquarium trade. Yearly variation in productivity of fish stock are function of age composition and growth pattern. Sustainable exploitation and utilization of Papiconum alpha as ornamental and fish food require basic information on the biology. Age and growth study and the derived growth parameter are indispensable for optimal and rational exploitation of a fish. We report age and growth of Papiconum alpha from Lekki Lagoon, Lagos, Nigeria. The study area is Lekki Lagoon, one of the freshwater ecosystem in southwestern Nigeria, where Papillomacosis alpha has been reported to occur all throughout the year. Month samples of fish were done for 24 months from landside of fishermen from Lekki Lagoon. The specimen was identified using standard guides and conveyed in eye chairs to the laboratory, where morphometry measurements like total length, standard length, and weight were done. Two approaches were used for aid and growth studies, and those are the direct method and the indirect method. The direct method is based on identification, interpretation, and photomicrograms of anal pattern, that is growth marks on art structures, namely sagittal otolith. Key scales were used, clitera, and opercular bone. Linear relationships were established between standard length and art part dimension. Back calculation of length and various ages according to Fisher-Lee equation were also established. The indirect method is based on analysis of length frequency distribution. The mean length as cohort were used to establish the formalizing growth parameter according to formalizing and Golden and Old plots. Growth derived parameters like lifespan, exploitation ratio, growth performance index, and total mortality were also established for the species. Data were analyzed using descriptive statistics in Microsoft Excel 2007. PSC correlation coefficient was used to establish linear relationship between parameters. Fish-based length analysis wizard was used to validate mode for length frequency distribution. Result and discussion, the indirect method. The length frequency distribution revealed 4 modes, and maximum age from this was 3 years. The most technical size of it was 4.5 cm, while the length at maturity was 41.1 cm. Preponderance of immature fishing, which constitute about 68% of the catch, suggests growth overfishing. According to Cauchin 1977, overfishing could occur in 2 forms. It could be growth overfishing or recruitment overfishing. Growth overfishing implies excessive harvesting of immature fishes, while recruitment overfishing implies excessive harvesting of reproductive stocking biomass from a population. In an unfished or well-managed fishery, the peak of the yield biomass is close to the optimal yield. But what we have here is that the most critical size is 30 cm, which is less than the less at optimal yield 49.5 cm, indicating growth and potential recruitment overfishing. The fertilizing growth coefficient value of 0.33 per year was less than 1. This shows that the species is relatively long life. This is also supported by estimated lifespan of 9 years. In fact, the inverse proportional relationship between acetonic length and fertilizing growth parameter suggests reliability of our estimate, and as a result was comparable to the growth parameter estimated for this species in other studies. The growth performance index of 3.29 suggests stability of this species for culture. The expectation ratio 1.9 is close to 2, signal over expectation, possibly due to increasing demand associated with white collection from the wild. A fish that approaches ultimate length quickly as it has a high value of K is likely to have high natural mortality. In contrast, a fish that goes slowly that has a low value of K is likely to have low mortality. Hence, mortality of Papillomacosis alpha in Lekki Lagoon could be more of fishing mortality since it approaches maximum length slowly because of low K value. The maximum age determined from direct method was 3, which is agreement with what we have in length frequency distribution. There were no clear annuli to facilitate aging from otolith, but we have well-defined and clear annular growth marks on other art structure like scale, clitera, and opercular bloom. Point correlation was established between scale radius, clitera, and opercular length with fish length, and the estimated length and back calculation, estimated ages and back calculation length were reported also. Limitation of the study. Uneven size of monthly sample. Selectivity of sampling J. Time period of deposition of growth mark were not verified. Conclusion. Preponderance of immature fishes of length size 38 cm in the car suggests growth overfishing. The aforementioned coupled with high exploitation and slow growth rate, indicated vulnerability of Papicona alpha to overexploitation. Papicona alpha in Lekki Lagoon, Nigeria, is exposed to exploitation and the need for its conservation in the lagoon. The recommended management ratio is minimum legal length of 51 centimeter. Minimum legal length indicates the size of a fish that can be removed from a population without impairing the reproductive potential of that population. And based on our study, this size is 51 centimeter. References. Thank you for listening.
Yeah, thank you very much, Oluwale. Yeah. We invite questions from our participants, please. And Oluwale, please, are you around?
Here, please. Hello, everyone. I'm here, please.
Right, great. Welcome.
Thank you, please.
Our comments and questions, please. And we didn't see any color picture of your knife fish because being an ornamental fish, it would have been beautiful if we had seen a picture of it. That would have maybe Ghanaians would have also embraced it.
The picture is there in methodology. The picture is there under methodology.
Okay.
It's there.
I missed the picture.
You missed that part, please.
Okay.
Thank you.
I'm going to stop his video because his network is not so strong.
Right. Are we okay? Right.
Yeah. Hello. I think there's a question in the chat.
Okay. Let me just read that. Chat. Can you please read it? How long was the frequency?
The sample was done for 24 months. A monthly sample for 24 months. That's a period of 2 years.
Sampling. Okay, monthly sampling for 2 years.
Yes.
24 months. Right. Thank you. Any other comments or questions? All right. I think thank you very much, Oluwale. We'll move on to our next presentation on effect of frequency.
I thought I did sit down there. I will just
We'll move on to our next presentation, please.
Hello everyone. My name is Christian Labi AEC and I'm presenting excerpts of our study, Effect of Feeding Frequency on Growth Performance of Juvenile Migrant Catfish. This study was conducted with Grace Deborah and Professor Elliot Haruna Al-Hassan. Introduction. Feed is generally the highest variable cost at aquaculture facilities. Therefore, understanding nutrient requirements and implementing appropriate feeding strategies is important. Feed management in terms of optimization, that is feeding rate and feeding frequency, has become a crucial area of study in the culture of many aquaculture species. A proper feeding practice involves providing a cost-effective foodstuff at the right times, in the proper amount, and in the proper form for the optimal growth of the fish. In aquaculture, both overfeeding and underfeeding can be detrimental to the production of fish. This is because overfeeding can deteriorate water quality, whilst underfeeding can lead to poor growth performance or starvation. Some studies have been conducted with respect to effect of feeding frequency on growth performance. feed utilization and immune response in several freshwater fishes. That is Okeke et al. 2016, Okomoda et al. 2019 conducted studies in Clarias garriapelos, whilst Alemahu and Getahun 2017 conducted studies in Nile tilapia. Until now, no study has been conducted with respect to effect of feeding frequency on growth performance, feed utilization, and proximate composition of bag-grade catfish. The objective of this study is therefore to evaluate the effect of feeding frequencies on growth performance, feed utilization, as well as water quality. Experimental design or experimental setup. The fish for this study was obtained from the Volta Lake, and they were transported to S-Point Fish Farm, where they were acclimatized for 1 month before the experiment. The fish had initial weight of 24.35 grams. That is the average. The acclimatized fish were then randomly distributed into 12 tanks. That is 4 groups with their 3 replicates. Each tank, with a size of 3 meters by 1 meter by 1 meter, was stocked with 30 juveniles of bag-grade catfish. Feed and feeding. They were then fed commercial diets purchased from Aquaculture, Ghana, with 58% protein and 17% fat. Feed and feeding continuation. So table 1 shows the times at which the fish were fed. For treatment 1, they were fed 12 noon each day. For treatment 2, they were fed at 10 and 4 p.m. each day. For treatment 3, they were fed at 8 a.m., 12 noon, and 4 p.m. For treatment 4, they were fed 8 a.m., 10:30 a.m., 1 p.m., and then 3 p.m. Sample collection. After the 56 days feeding trial, the weight and length of the fish were measured in grams and centimeters respectively. Then, in order to assess growth performance and feed utilization, the following calculations were made using standard formulas. Weight gain, specific growth rates were calculated. Also, condition factor, feed intake, feed conversion ratio, protein efficiency ratio, as well as percentage weight gain were calculated. Water quality parameters were also analyzed following standard methods of APHA 2005 for nitrate, nitrite, pH, and ammonia, whilst Testo 110 was used to measure temperature. And then dissolved oxygen was also analyzed using Crescent Oxy 45 model oxygen meter. Statistical analysis. Data were analyzed using graph pad prism, and then the data is presented in graphs and tables using the mean standard error of the means. Results and discussions. Results and discussions. Growth performance. Final weight, weight gain, condition factor, and specific growth rate were calculated as stated earlier on. And the results showed that feeding bag-grade catfish twice a day, that is treatment 2, had the best final weight and was significantly higher than all other groups. This was same for weight gain, condition factor, as well as specific growth rate. Feeding bag-grade catfish twice a day performed better and it is in agreement to earlier reports by Karen et al. 2002, Doya et al. 2002, Ashley Dejo et al. 2014, as well as Marimutu et al. 2010. Feeding bagred catfish 4 times a day, however, recorded the least performance, and there are 3 major reasons why they might have performed poorly. 1. Energy expenditure may increase as a result of increased swimming activities arising from feeding throughout long periods of the day. 2. Food passing through the digestive tract more quickly, leading to less effective digestion. And then 3. Reduction in absorption efficiency resulting from gastric overload due to feeding at relatively short interval than the time required for the fish appetite to return. The lower growth rates recorded in fish fed 3 and 4 times a day could also be as a result of oxygen consumption as well as increased foraging activity. Effect of feeding frequency on feed utilization, hepatosomatic index, and viscerasomatic index. The present study revealed significant difference in feed intake, and this is in agreement to previous studies by Aderolu et al. 2009 as well as Gore et al. 2018. Feed intake is related to the capacity of the stomach, the rate at which feed is digested, as well as how fast food is evacuated. The suitability of feeding frequency is therefore affected by the interval between feeding. Fish fed twice a day recorded the best growth performance or the best feed intake and corresponded with the best growth performance. The implication is that feeding bagred catfish twice a day at an interval of 6 hours could enhance growth. Effect of feeding frequency on water quality. Dissolved oxygen and ammonia were significantly altered by feeding frequency. With dissolved oxygen, it was observed that the higher the feeding frequency, the lower the dissolved oxygen. And then with respect to ammonia, the higher the feeding frequency, the higher the ammonia content. It must be stated that all the parameters observed with respect to water quality were within the requirements for culturing of catfish. Then we did a correlation analysis between water quality, growth performance, and then feeding frequency. Feeding frequency correlated positively with pH and nitrate, but negatively with dissolved oxygen. There was no significant correlation between feeding frequency and other water quality parameters such as nitrite, ammonia, and temperature. In conclusion, feeding frequency affected growth performance and feed utilization in culture of bagred catfish. Higher levels of feeding frequency could deteriorate water quality by decreasing dissolved oxygen content and increasing levels of ammonia imports. Thank you.
Yeah. Hello. Hello. Thank you very much for your presentation. That's Christian. Yeah, so we invite questions and comments from our colleagues, please. Yeah, Oris Sasona. This time I got it right, I hope.
Yes, you did. That was better. Well, thank you very much, the presenter. I must commend your work. And even for the fact that you were able to procure from the wild, if I'm not mistaken, the juveniles of the bagrid fish you use for your work, and getting an initial weight that I guess will be uniform. That's really commendable. But I have a question, or maybe I missed out in the course of your presentation. Yes, I got the point, the frequency of feeding. You fed once, twice, thrice, like that daily. But at what rate? Were you feeding at a particular percentage of their body weight, or you were feeding to satiation? And for either of the two, how did you make the choice? And again, I want you to explain how you were able to calculate the feed intake, most especially if it was if you fed to saturation. Thank you.
Yeah, Christian. Is Christian or any of the group members?
Do you want to comment?
Deborah and Alasan, if any of you is available to answer the question. Unfortunately, our presenter is not available to answer questions. And so we would put that on hold once again when they are on, or if we are able to save time at the end of the whole session, we would come back to these outstanding issues. And so we'll move to our next presenter on water quality. So Aris, I'll say Aris now, and then Godfred, please hold on with your question. And then we'll move on to our next presenter on water quality sample integrity by Ebua.
I'm Doris Yewaba, presenting on the topic water quality sample integrity and implications for aquaculture development and practice. In recent times, the Ghanaian aquaculture industry has seen several improvements with fingerling quality and quantity as well as feed development. However, little attention has been given to water quality management, despite its effect on the productivity of the sector. Of production systems and the effect of effluent discharge on the environment are dependent on how water quality is managed within the system. There have been several efforts by some farmers and researchers to monitor and manage water quality in the systems, but where and how the samples are collected, how the samples are handled, as well as the quality of analysis and instruments used affect the quality of data obtained. Decisions and decision making at both the farm and research level are dependent on the quality of data that is collected. For the past 2 years, we have been working and managing a water quality lab, and our presentation focuses on the state of samples getting to the lab by highlighting some problems that are particular with chlorophyll A samples. The first problem is with labeling of samples. The common practice is that samples that get to the lab either come without labels or without specific IDs, or sometimes the labels are unclear or not readable. Some also come without dates. This makes identification of samples a problem. The recommended practice is for samples to be well labeled, that is the sample number or sample ID, as well as the location, the date, and the person who did the sampling using an indelible ink. Sample containers have also been a problem, as some samples come in clear bottles that exposes the samples to light, thereby resulting in continuous photosynthetic activity, which produces new biomass. The recommended practice is for chlorophyll A samples to be collected in dark bottles. Thus, the samples should be excluded from light. The preferred sample bottle for chlorophyll A samples are the amber bottles, but in cases where amber bottles cannot be gotten, clear bottles should be wrapped with either aluminium foil or nylon bags. Sample collection has also been seen to be a problem, as most of the samples or some samples that come to the lab come not being filled to the brim. This creates problems like the agitation of samples that produces gas buildups. It is recommended that the sample container is dipped completely in the water before it is opened, filled, and then capped still under the water. Storage and transportation has also been a problem. Some samples come to the lab with the whole sample being frozen. Others also come without ice or under warm conditions. This poses problems like degradation of samples, as well as continuous photosynthetic activities and increased biomass, which might not be a true reflection of the natural system. It is recommended that samples being transported to the labs are kept on ice. And then in situations where the samples cannot be analyzed on the same day, the sample is either filtered and stored in a dark or a non-reflective substance or container and kept frozen in the refrigerator, or the extracts stored at 4 degrees Celsius overnight. Another problem is also with coordination. Most often, people would not communicate with the lab technicians or people in the lab, but would rather on their own decide to pick samples and bring it to the lab. This poses problems with storage, as well as long or over storage of samples. This results in sample degradation. The results will be gotten alright, but they might not be a true reflection of the data of the system, the natural system. So it is recommended that researchers as well as farmers coordinate with lab technicians to find out the time that would be appropriate for their sampling to be done and when the samples should be brought, in order to ensure that there is adequate space for storage and analysis. Common issues apart from chlorophyll A samples include improper sampling for gases, that sample bottles not being filled to the brim. Example is with dissolved oxygen samples. It is recommended that sample bottles must be completely dipped and capped under the water. Also, storage of alkalinity samples has seen to be for several days before analysis. It is recommended that samples are analyzed within 24 hours after being stored at low temperatures. Biological and chemical oxygen demand samples are also usually exposed to light, and that should not be the case. It is recommended that samples are collected in dark bottles and kept away from light. Wrong and long-term storage of nutrients, example nitrogen analysis samples, is also seen to be another common practice. It is recommended that Samples should be transported and stored under low temperatures. This is because the various stages of nitrogen in water changes as time and temperature changes. So it is recommended that the samples are analyzed within the shortest possible time. Quality monitoring and management requires as much attention as all other aspects of the industry. This is because it is an important factor as far as aquaculture development and advancement is concerned. And to achieve this, quality and accurate data is required. It is therefore important for all stakeholders to prioritize this by training in terms of sampling and handling of samples, and the use and interpretation of data so as to achieve sustained growth. We would like to acknowledge Professor Daniel Ejeboating, the project coordinator for IPAC projects from the Department of Fisheries, KNUST, for the opportunity given us to work in the lab. Another thanks goes to Mr. Stephen Jemfi for the pictures. Thank you for your time.
Yeah, thank you very much, Doris. Yeah, so once again, we invite questions from our participants. Please.
Is Doris online?
Questions and comments and contributions, please. At least for many of us, this presentation cuts across a lot of the things we'll be doing, the data we collect in our everyday life. And so for all of us here, I think some comments, contributions, questions will be useful. Yeah, Godfred. Please, Godfred, unmute yourself and ask your question.
Thank you very much. And I would like to ask, she didn't state the type of water she's dealing with, like whether it's borehole, tap water, rainwater, or whatever, like in case of water sampling.
Yeah. Yes, Doris. Yes, Doris.
Yes, please.
Your question.
My focus, yes. Hello, please. Please, can we hear me?
Yeah, I'm listening.
Okay. So the focus is basically on aquaculture. So mostly is with pond water systems. And then sometimes too, some farmers come with samples like their borehole water they are now going to stock their ponds with. So, but for this presentation, the focus was basically on chlorophyll A samples. That is when the fishes have already been stocked. Thank you.
Okay. Okay, right. Thank you.
Right. Yes. Any other comments and contributions, please? Okay, and so we move to our next presentation. That's on next. Yeah, that's water exchange. Water exchange method. Oh, okay, Collins, your hand is up. Before the presentation, a question from Collins.
Okay, yes. Thank you very much, Doris, for that wonderful presentation. I think it is very relevant. But I want to ask for farmers who may not know how to sample water from their ponds, how to keep water from their ponds. How have you have been in the lab? So how do you get this information to the farmers that the way they carry their samples or the way they bring their samples to the lab is not the best way? Have you had any opportunity to educate them on how they should do it in the future?
Okay, thank you very much. Please, with the farmers that come themselves, mostly I educate them on the way they should sample next time. Sometimes some of the samples are even rejected so that they collect in the right way. And I also think going forward, what can be done is for the Fisheries Commission and all other stakeholders in the aquaculture industry to collaborate and train farmers, since we know what is the best practice or what to give quality data. We should work and train the farmers on how to do proper and accurate sampling. Thank you.
Thank you.
Much. Yeah, if no hand is up, we move on to our next presentation. Our next presentation, please.
Hello there, good morning. My name is Shofela Shofalabi Olaniyi. I will be presenting to you today findings from our little trial, which we've titled Water Exchange Method Affects Feed Consumption and Feed Conversion in Clarias garapinos raised in small plastic tanks. Here's a quick overview of my presentation today. Alright, so introduction. A common practice among many catfish farmers, regardless of the kind of rearing system they have, is that they constantly exchange water. And water exchange is extremely important for optimal growth and even survival of catfish because it helps to get rid of solid waste. And this is particularly important in Clarias because they are hair breeders. Therefore, the carrying capacity or production capacity of the rearing tanks they are being held depends more on how much waste can be washed out or recycled as opposed to oxygen levels. So in other words, unlike many fish species, a buildup of ammonia and nitrites is more likely to impede growth of catfish, Clarias, than oxygen levels. Therefore, getting adequate information on how best to exchange water is very important. But much of the information available on the efficacy of the methods of water exchange were at best guesses with very little or no scientific backing. And although we had some adequate information about the amount of water required to be exchanged for a particular density of fish in a tank, the question we sought answers for was how best to go about it. That is, should it be a steady flow through of water or a one-time exchange? And if it's a one-time exchange, how often? Hence, we decided to test 3 common modes of water exchange to see if there will be any difference in feed consumption or feed conversion, so we could better design future trials and for comparing feeds or comparing different rates of water exchange. So, but in general, this information is very useful for anyone interested in growing catfish. So a quick run through the materials and methods. This trial was conducted in a farm in Ibadan, and 12 plastic tanks with carrying capacity of 1 cubic meter were each stocked with 25 Clarias garapinos catfish and divided into 3 treatments. Each treatment had 4 replicates, and the daily equivalent of 500 liters of water was exchanged in all treatments in 3 different schedules. So each water exchange schedule represents a treatment, and in treatment 1, water was exchanged by reducing the entire volume of water in the tank by 50% and then topping back up to the initial volume in the tank. In treatment 2, water was exchanged by entirely emptying the water in the tank every other day and then refilling the tanks. So in treatment 3, 500 liters of water was slowly released into the rearing tanks and allowed to flow through. So on to feeding. Feeding was done once daily and in the afternoon, and with feed of crude protein 42% for 87 days and 33% crude protein for the remaining 28 days. Dissolved oxygen and temperature were measured every day just before feeding, and weight sampling was carried out every 2 weeks. So statistical analysis was carried out using RStudio. So the data obtained from this feeding trial was analyzed using one-way analysis of variance while the mean differences between treatments was tested for significance using Duncan's multiple range tests. So this is the experimental setup, and one thing to note here is that all tanks drained water from the bottom, and so to your left, if you see to your left, there are some white header tanks which were used to hold exactly 500 liters of water, which were slowly released into treatment 3 for flow through. Here are some of the growth performances of the treatment, and if you look at the final weight, you discover that both treatment 1 and treatment 3 performed better than treatment 2. Survival was high across all the treatment, and some of the mortalities we had were as a result of fish jumping out. Although we had meshes pre-installed to prevent this from happening, but some of the fish still managed to escape. So, in other words, if the mortalities were in the final weight would have increased a little bit, but that's not very significant. So, but the specific growth rate confirms that both treatment 1 and treatment 3 are the better treatments. So, in general, from the growth perspective, the treatment with a one-time 500 liters daily water exchange and the continuous 500 liter per day treatments were better. So, in terms of feed utilization, fish in treatment 1 and treatment 3 consumed more feed. And during this trial, both treatments responded to feed very well in comparison to fish in treatment 2, where the response to feed was notably low. And fish in both treatment 1 and treatment 3 also converted feed better. FCR was high in this experiment, mainly due to the slow growth rate in the subadult phase, which is the phase of the claras we used in this experiment. In other words, the FCR data is bound to be a little bit high, since the faster fingerlings growth rate phase has not been taken into account. And another reason I can think about for the high FCR is the fact that we had some mortalities, which I explained in the previous slide, especially in treatment 1 and 3, and it could have helped to reduce FCR a little bit if it was accounted for as production. Anyway, the results still helped to separate the treatments according to the stated objectives, which was to test the best mode of water exchange. So to reinstate, changing water using the mode of water exchange in treatment 1 and treatment 3 led to better feed conversion ratios. Again, in terms of the economics of the treatments or the modes of water exchange, treatments 1 and treatment 3 would pass on some savings to a catfish business. Because the prevailing price of catfish in Nigeria right now is around 700 naira per kg, and a business setup using treatment 2 as a mode of water exchange might end up in losses. And using the volume of water used per kilo of flesh gained, treatment 1 and treatment 3 again will pass on some savings to a business, a catfish business, because they were more efficient water users. So imagine a business using diesel to pump water. The cost of water would also would almost double if it was using treatments, the mode of exchange of water in treatment 2 as its primary water exchange method. So this graph shows that treatment 1 and treatment 3 were very similar in terms of weight gain throughout the experimental phase, and also that treatment 2 began to show signs of poor growth as early as 4 weeks into the experiment. So it was evident that early on in the experiment that treatment 2 was an unsuitable means of water exchange to the fish. Dissolved oxygen and temperature readings were taken during the day around noon time. And the most important thing to note from this table is that dissolved oxygen DO was very high in all treatments. And in spite of the overwhelmingly high DO, treatment 2 still performed poorly in terms of growth and feed response. In conclusion, treatments 1 and 3 were better modes of water exchange in terms of feed response, growth, and feed conversion. Even though similar amount of water was exchanged in all treatments, a small, more frequent exchange worked better for Clarias production. It is recommended to avoid abrupt changes in water quality with Clarias, and this trial actually measured how much of a disadvantage full water exchanges can be, even if bad water is being replaced with good. Removal of solids seems to be key, and it's known to be of high importance in Clarias production. Lastly, in terms of the environment, the wastewater discharged from a fish setup, such as in this trial, is very full of nutrients and therefore should be disposed properly. And for example, it makes good fertilizer for plants, especially vegetables. And we were not able to fully utilize it during this trial, but we were sure to dispose the effluents responsibly. Lastly, we are grateful to the American Soybean Association and the World Initiative for Soy in Human Health, as well as Top Feeds and Kinset Agro Services for jointly funding this project. Thank you very much for listening.
Yeah, thank you very much, Sofela. Right, so that was a presentation on water exchange methods. Please, once again, we invite questions, comments, contributions. Nuruddin, you are welcome. You can ask your question. Unmute.
Thank you very much, sir.
Ask your question.
All right.
Thank you very much, and I'm very happy and glad to see this presentation comparing the mode of water change, and it's just to applaud the experiment, because this is what most of our farmers in Nigeria have been asking questions on which method to use, and now that we've gotten an experiment with data to show which one is better, I think it is good to go for catfish farmers in Nigeria and across Africa to know that it is not actually ideal to change all the old water, but it is essential that we change the water. But I just want to ask, sir, that the experiment, because we know the normal practice in Nigeria is that you see farmers changing their water maybe after 2-3 days, maybe because of economics, electricity, and stuff like that. But this experiment did not inform whether this exchange of water flow through or 50% should be done. What frequency? Is it daily? Is it once in a month or once in a week? I think it will have been good to guide our farmers right concerning that.
Okay.
Thank you.
Thank you very much. Thank you very much. I think it's a very good question, a very brilliant one. Concerning the frequency, for this experiment, it was a daily exchange. I hope I'm communicating. Can anybody hear me?
Yeah, we can hear you loud and clear. Carry on.
All right, great. Thank you. So for this experiment, it was a daily exchange. I mean, for a practical setup, I will, for example, in this experiment, the water still expended for this experiment per treatment, I would say is still I would say a little bit more than what I would do if I were doing it practically. For example, in the earlier stages of the experiment, I would practically will have changed the water less frequently. I mean, not less frequently. I would change the volume of, I will change little volume compared to what was being changed in this experiment. But for experiment's sake, for standardization for the number of days, we had to use the same quantity of water, daily. But to answer your question, it was a daily exchange. But for practical sake, it's left for the farmer to work out, the quantities or the frequency of exchange the person wants to do. So, but for me, I would say we did a daily exchange.
Right.
Thank you.
Thank you very much. And next, Collins, unmute yourself. Your question, please.
Yes, okay, thank you very much. I just want to seek a clarification about something. With the treatment 3, where it was a flow-through system, he indicated in the results the volume of water that was exchanged. I want to ask how the measurement was done. If it was a flow-through system, how were they able to measure the volume of water that went through that system?
All right, great question. Again, very brilliant one. If you check one of the figures that the pictures that I put in the slide, you would see that there were some header tanks, towards the right, and that's what I was trying to show. because for the treatment, for the flow-through treatment, we had to think about how to get exactly 500 liters of water through being flown through. So what we did, because the white tanks that you see in the picture, they are also graduated. So that means they have the 100-liter mark, 200-liter mark, 400-liter mark. So for example, for treatment one, changing, halving the water in the tank was very easy, because you just had to reduce the water to 500 liters and then topping back up. And for the flow-through system, what we did is that we cut that white tank into two and made a scaffold for each of the treatments. So every morning we pump into half of that tank, which we splitted, and then we allow it to slowly trickle into the system. I mean, I don't know if you understand what I just described, but it was very, very, that way it was easy to measure without, going back to check, and withstanding the risk of, over exchanging too much water per day.
Right, thank you. Now, Godfred, your turn, please. Godfred, unmute yourself to ask your question.
Thank you very much. With treatment 3, you said you exchange water daily. Do you,
yes.
Is it a recirculation system, or you allow the water to be disposed and then you put a new one? And if it is a recirculation system, how often do you dispose of the final water?
Okay, so this isn't the recirculatory system. It's just a flow-through system, and it's just a simple flow-through system, which we all know. For recirculatory, it will have been, I will have stated in the methods, but it wasn't, and it was clear in the methods. If you followed the methodology, it was a flow-through system, so it isn't a recirculatory system at all.
Right. Thank you. And now, Adafi Odioko.
All right. Thank you. Thank you. Adafi Odioko is the name, from Nigeria. We're presently a PhD candidate and then investing in Turkey. Yeah, I want to thank you for the wonderful presentation. Coming from the economic aspect of it, did you consider the cost of electricity for the three different methods? We know the situation in Nigeria, where electricity is not stable, and if one have to use a generator to power this thing, so do you consider the cost of electricity in your economic calculation?
No, I didn't consider my own cost of electricity, but if you were to apply this to yourself, I would advise that since you already know the volume of water that was exchanged, it's better you just calculate, per meter cube, how much it costs you or the farmer, if I may put hypothetically, how much it costs the farmer to pump a cubic meter of water, for example, and then multiply. That gives you the economics. But for this particular research, I didn't do the economics because I kept on fluctuating from gen, to pumping using electricity, and even during the experiment, electricity tariff got changed. I mean, like almost. I mean, I can remember once. So I mean, it's just better I just leave it that way. I hope I answered your question.
Right. Thank you very much.
But to you, which of them will cost more? But to you, which of them will cost more money? For the flow-through system, do you need electricity to operate the system? And if yes, do you think that will cost more money?
Okay, you mean to operate the system. For example, the flow-through system, I mean, if you are pumping 500 liters of water into treatment, for example, treatment 1 because we're talking about treatment 1 and treatment 3 being the better treatments now. So if you are pumping 500 liters of water into treatment 1, it's also the same thing as saying pumping 500 liters into treatment 3. Because once you pump into your storage tank, it's more like it flows by gravity. I don't know if that answers your question now. So, it flows by gravity. Yeah.
All right, thank you. I thought it was powered by electricity. Thanks.
Right. No.
Thank you very much. So we now move on to our next presentation. That's from Afrimakwa. Yeah. Afrimakwa is a research network for sustainable marine aquaculture in Africa. Yeah, marine aquaculture, that's quite a rare opportunity for Africa. Let's hear what they have for us. Afrimakwa, please, our next presentation.
Good morning to everyone, and thank you very much to the organizers of the conference for allowing me to present you today the Research Network for Sustainable Marine Aquaculture in Africa, Afrimakwa. Aquatic organisms are valuable sources of macro and micronutrients as they contain high-quality protein and essential amino acids, omega-3 long-chain polyunsaturated fatty acids, and essential vitamins and minerals, so they play an important role in human nutrition. As you all know, global catch has stagnated for the last 3 decades, and aquaculture has grown rapidly during this time to meet the growing demand for aquatic food, and today more than half of the consumed fish comes from aquaculture, a percentage that is expected to further increase by 2030. Indeed, aquaculture will play a major role in future food production and will be essential to contribute to global food and nutrition security. In Africa, unlike in the rest of the world, seafood consumption is not expected to increase by 2030 due to population growth outpacing supply on this continent. Indeed, Africa has the fastest growing population in the world, and as you can see in this image, it will account for nearly half of the global population growth over the next 2 decades. In addition, the most commercially important fish stocks in Africa are fully or overexploited, which is the main reason why Africa has become a net importer of fish for almost a decade now. And although the annual growth rate of aquaculture production in Africa has been the highest throughout the new millennium, it still represents less than 3% of the world's total production. As you can see in this graphic, both brackish and freshwater aquaculture production have greatly increased over the last 2 decades, while marine aquaculture production has remained relatively low. In 2019, brackish water aquaculture represented more than half of the total production, followed by freshwater aquaculture, and marine aquaculture only contributed to 5% of the total African production. It is also interesting to note that almost the totality of the brackish water production is represented by fish, whereas seaweeds dominate the marine production, followed to a lesser extent by mollusks and fish. So there is a general consensus that marine aquaculture has great potential for development in Africa. However, as you have seen, it is far away from the existing production in brackish and fresh waters. There are multiple reasons behind this, such as conflicts of resource utilization, energy, quality of seeds and feeds, feed prices and availability, product standardization, marketing and trade, adequate policies and framework allowing the development of the sector, but also lack of expertise or insufficient research and innovation. And in regards to the latter, Afrimakwa, funded by the French National Research Institute for Sustainable Development, was created in 2019 to gather researchers to facilitate scientific cooperation for the development of sustainable marine aquaculture in the region. The network is currently composed of 19 institutions from 6 African countries: Kenya, Tanzania, Mauritius, South Africa, Namibia, Côte d'Ivoire, and Senegal, and also France. So the ultimate goal of this network is to contribute to the development of sustainable marine aquaculture in the continent, to combat poverty, ensure food and nutrition security, provide jobs, and create income and wealth. The main objectives of Afrimakwa are to share knowledge between partner countries, to reinforce the capacities of the consortium members, and to develop joint research projects. And to do so, our activities include training workshops, short-term scientific missions, e-learning, meetings and webinars, and joint publications. With these activities, we particularly aim at promoting the integration and active participation of students and young researchers within the consortium. We also ensure the dissemination of the results produced by the consortium to scientific, governmental, and industrial actors, as well as to civil society. The research network articulates around 4 main research interests: the control of the life cycle of aquatic organisms of commercial interest through the development and/or improvement of breeding protocols, the development of high-quality and sustainable feeds, including the use of agri-food waste, microalgae, or insects as alternative sources to fish meal and fish oil, the development and improvement of eco-friendly farming systems such as integrated multi-trophic aquaculture systems, and the improvement of the welfare and health of organisms and farming systems, including risk assessment and alternatives to the use of antibiotics, strengthening of the immune system to fight against pathogens, or the control of water quality and other welfare parameters. In Afrimakwa, we study a variety of organisms, including marine and brackish water fish, crustaceans, mollusks, holoturians, echinoderms, corals, and algae. Here I will show you some examples of research actions that are currently being developed by Afrimakwa members in West Africa. For example, in Ivory Coast, the oyster Crassostrea grassar constitutes a valuable source of protein, and the artisanal harvesting of this oyster represents an important source of income for coastal populations. In order to master the breeding of this species, our partner Dr. Blair and his colleagues are studying its biological basis and more specifically its reproduction cycle. They have found that the maturation of sex cells occurs in 5 stages, that the sex ratio is in favor of females, and that the reproduction is continuous and closely linked to the physico-chemical conditions of the environment, in particular the salinity. They consider that both the continuous sexual cycle and the sex ratio in favor of females are positive characteristics towards the development of the culture of this species in Ivory Coast. In Senegal, efforts are currently being made to promote brackish waterfish farming, in particular with the West African tilapia Sarotherodon melanotheron, a very resilient species which is erytherm and euryhaline. An example of this is an aquaculture farm located upstream of the Saloum estuary in the Fatigue region. However, yields are not very good, and our colleague Dr. Tine is currently testing the effect of stocking densities to help improve production. And the next example of research is being conducted in France, but in cooperation with a company in Senegal, and also deals with the West African tilapia species I just mentioned. Taking advantage of its detritivore feeding habit in adulthood, we are studying the nutritional needs of these species cultivated in an integrated multitrophic aquaculture system, combining bacteria, phytoplankton, and fish, allowing to recycle the waste produced in the system. The objective of this study is to develop a feeding protocol that reduces as much as possible the production costs. Finally, another project that is currently being developed in Senegal by our colleagues Dr. Ndiaye and Dr. Diadiu and their partners is focusing on the study of the taxonomic and genetic diversity of seaweeds, their biodiversity and habitats, and their environmental stressors. The ultimate goal of this project is to provide a roadmap to policymakers to encourage both exploitation of seaweed and preservation of natural seaweed habitats as a tool for fisheries management and as a refuge and habitat for exploited fish. And last but not least, I would like to highlight that Afrimakwa is open to other African institutions, and you are more than welcome to contact us by email or to follow us in our social media accounts. Thank you very much for your attention.
Hello. Yeah. All right. So thank you very much. And that's a nice presentation from Darius. At this stage, we invite questions, contributions, comments. This is an Africa-wide initiative. And so if you know of an initiative elsewhere that hasn't been captured, please, you can bring it up for discussion. And this comments and contributions. Yes, Rhoda.
Yes, thank you very much. I think that was a very good presentation, and I think I am really liking the kind of job, like the amounts of work they are doing. But I noticed, unfortunately, Ghana was not one of their focus countries. I don't know why, but then I also want to know, I'm not sure whether I missed out, but I want to know what she thinks is the actual challenge in going into mariculture in West Africa. What the actual challenge is, because it's really not done per statistics, it's shown that it's very minimal. So what is the actual challenge in going into mariculture in West Africa? Thank you.
Thank you very much. Do you hear me?
Yes, we can hear you loud and clear, Darius.
Okay, thank you. Good morning to everyone. Thank you, Rhoda, for your question. Indeed, the difficulties of marine aquaculture development are very, very extensive and different between regions. So I cannot tell you one or two reasons for West Africa, but what I know is that there are some questions related to the use of the environment, like conflicts of use of resources, and between other sectors of the industry and even tourism, and also there are things related to the policies and frameworks that are not in line with the possibility for farmers to develop their companies. But of course, if you would like to know more about that, I can put you in contact with colleagues from different countries from the West Africa specific regions, so they can really detail you more about the specificities of the problems in that region.
That if your network is pretty good, you can enable your videos so we can at least appreciate.
All right, and I'm sure there will be a lot of questions. Why not Nigeria? Why not Ethiopia? Yeah, but Godfred, your comment or question, please.
Do not, because it would sabotage.
I would like to know why mariculture is not really a practice within the Western African waters. Like in South Africa, there is some aspects of mariculture. I don't know of Egypt areas, but within Western Africa, Nigeria, Ghana, Togo, those waters, I think it is not viable because of the distance from land to the far seas. If she can explain further, since she's done research on the African waters.
I'm sorry, I didn't hear very well the whole comment, but what I also
I am asking that why is it that there is no mariculture practice in the West African waters, like Nigeria, Ghana, Togo, Ivory Coast, and the rest, but there is much practice in South Africa and some parts of the oceans? Yes, is it because of the distance from land to the far seas? What is the main reason? Sometimes they say it is not viable economically.
Yes, that's the main. Yeah, yes, there is some technicality problems that are a common problem in marine aquaculture, in general, and also in your coasts. We know that we have to find some kind of some kind of aquaculture systems that are not as technical as we should use in some of the cultural species, like with RAS circuits and all this stuff that are very, very energetic consuming. But to use some of the tides, the use of the tides of the sea to renew the water, and so you have to have some kind of areas
available to do this kind of aquaculture. I am thinking about fish aquaculture, like tilapias, for example. But the problem of marine aquaculture is that it's very expensive compared to freshwater aquaculture.
Okay, but if I may ask, do you consider bringing water to the land and then to give it the same atmosphere like what you have in the sea, to do a practice on the land with mariculture, like bringing water to the land and then it is like we're doing the same stuff in the sea, not necessarily going to the sea, but on the land?
Yes. Well, that also depends on the quality of the coast. If there is the possibility, for instance, in the south of Africa, there is very difficult to have aquaculture in the water, but you have to bring inshore, because inland because it is very difficult regarding the currents. But it depends on each country, because if you can have some places in the sea that are suitable for marine aquaculture offshore, that is a possibility, but it's very expensive. So that's like a handicap. Then indoor, it's also more, I would say, more sustainable, but it has also other costs associated. So it's difficult to respond to such a question, because it depends on many other factors around. It's a multi-actor issue that has to be analyzed as a whole to see what is the better situation for each region.
Okay, right. Okay, thank you.
Yeah. Any other questions and comments? Please, now, can we, those who missed out on the questions when they were not available, we are open for any comment, questions on aquaculture. We've had presentations on diseases. We've had presentations on limited to tilapia, catfish. We've heard of water quality. Yeah, and now we've heard of something about mariculture in general. So if we can tie up all this and then have a general discussion on the aquaculture industry in Ghana, not in Ghana, sorry about that, in Africa, with respect to diseases, the challenges, we can build up from yesterday's discussions as well. Yes, Nuruddin.
Thank you very much.
Yourself.
Thank you very much. It was just on the question that we wanted to ask during the presentation about the man from Ghana presented. Sorry, I missed the name, concerning the feeding, and then it was not available then. It's just to put an observation there. There was a place where he said they fed 53 grams of juvenile that were being caught and put into tanks. And they mentioned the feed, aqua feed, that was being used 58% protein and 17% fat. I think we need more clarification there, maybe for the paper, because that feed at that age is meant for juvenile that are just about 5 to 10 grams or maybe 15 grams. So at 53, the feed should be consuming 2 millimeter, which are 45% good protein. And so it's just an observation, and maybe possible, maybe it was a mistake there. So just the correction. And speaking largely about generality of aquaculture in West Africa, I think
Can you please hold on? Let me ask if the presenters are available. I see that's effects of feeding frequency on growth performance of juvenile.
No, not that one. The one before then, it was a presentation from Ghana, and they were talking about feeding.
From Ghana.
Yes, they were talking about. Sorry, so I'm trying to bring the screenshot. It was the one where the experimental design where the feeding fish were fed commercial diet from Alakwa, and the experimental design was a acclimatized fish with round. Okay, let me get the topic and
yeah.
Okay, where the objective was to evaluate the effect of feeding frequency on growth performance
that's exactly the one I'm talking about.
Oh, good. Oh, good.
Yeah. So IEC, and then Deborah and Alasan, if you are available. Is any of you available now? IEC, Deborah, Alasan. Yeah, but please, if you could, after this comment, you could just put it in the chat, and then
okay.
we can pass the comments to the authors. That might be very useful for their reviews.
Okay.
Yeah. So
then,
now let's
generally on the aquaculture and in West Africa, I will want us to have this continuous program like this to give awareness to farmers. I'm sorry to say, because if you look at this program that we have today, it's most of a scientific. The educational practitioner, I can hardly count the few numbers of farmers that are joining this program, and that means we need to take this awareness and this research we are doing to them, because the research is not useful if we keep it only to ourselves, the academia or the commercial partners, not the farmers. So we need to take it to them, because that is the only way. That means we need the extension services among us to give it to them so that they can be to have impact on the fish farming, and that is the way to go. And lastly, that our farmers start seeing fish farming as a commercial business, not a part-time business or a subsistence business. They should see it as a full-time commercial business that is multi-billion dollars and that can sustain our food basket and give us the leaps growth that we need in Africa and at the world at large. Thank you.
Yeah, thank you very much, Nuruddin. Yeah, I think we have a few farmers online. My knowledge, I have seen a few farmers, but I think there's still room for more farmers to participate. And some of us are also working we collaborate with farmers, we do research on farms, and therefore there's the need for us to carry this feedback to our farmers. So don't just go to the farms as a point of collecting data, but through collection of data, let's bring some of these results that have been presented here, the findings that have been presented here should also, we act as extension agents. Thank you very much. Yes, any other? As I said, it's open now on aquaculture in general. That's Godfred, yes.
Thank you very much. Actually, I think that aquaculture is taking a lot of stance in Africa as a whole. But with regards to, yesterday's discussion, all the way to today, we spoke about water quality, water exchange, feed, the cost of feed, and all those things. But is there any funding available for the practice? Because I have started aquaculture since 2008 in Ghana, and I've not received a single funding for my project. My farm is very big. They come to the farm to do research, they pick data, but I fund my farm with my own money till now. So for us to progress, to solve the problem of nutrition and unemployment, is there any program to help the farmer? Because sometimes you give money to people who have not even started farming and they misuse it. But we have the farm, in my farm, I brought light to the whole village, 90 post-2 transformers. You see, and I powered the whole village with my own money. I fenced my farm. I have my own dedicated transformer with my accommodation, everything there. But who thinks about the Ghanaian farmer? Who thinks about the African farmer? And we pump money into research with no results. But why want to develop what we have? Catfish, tilapia, and a few. We import a lot of fish into Africa from other countries. How do we help the African farmer to start to produce? We have got the space and the waters to do that. But we pump money into research on the same type of fish, tilapia and catfish. We don't need any other research on producing tilapia and catfish. We don't need it. We don't need any new, because we know how to produce from fingerlings, fry up to whatever, and to improve their weight. And as you are showing us on this program, we've realized some lapses, and we are going to upgrade. But is there funding for this? Is there funding for us to increase production?
Well, right now they are going.
That is my question now. That is my advocate.
Yeah.
We're joining several trainings, but we don't really help the farmer to produce fish. And we import a lot of fish into Africa. That is my question.
Right. Thank you. I think this is the cry of a farmer, and out of the pain of a farmer, he says we don't need further research on it.
We don't need it.
The main thing is that let's diversify the research focus. Let's broaden the research focus and try and bring on board other culturable species. So the farmers are crying out to the researchers, please, let's see what are the other culturable species. But of course, let's also see how we can improve on the production, the quality of the tilapia and catfish as well. I think the profitability of the catfish and tilapia, we haven't reached the ceiling yet. So there's still more room for improvement whilst we diversify the research focus. Yes, please. Any other comments and contributions? Yeah. Oladosu.
Yeah, thank you very much. Can you hear me?
Yes, we can hear you loud and clear.
Good. I wasn't part of yesterday's discussion, but with what we have on ground that we are discussing now, I think I would like to have an input in this direction. Yes, we have done so much on clarias and tilapia, but we find out that over the years we are having problems, especially on the health of cultures. Hatcheries are having problems. In fact, some are closing down because of incidents of diseases, and this is affecting seed propagation. And of course, you will agree with me that this is multifaceted. It's not. It has a lot of. We have various causes. We have various factors that are responsible for this. We have terrible inbreeding, depression, which is a big problem. We have serious upsurge of emerging diseases, which we need to focus on and ensure that productivity is optimal. Not that we have not perfected the art of production, science of production, but we are having problems that are hindering productivity, which we need to address. Farmers are abusing antibiotics so seriously. In fact, even researchers are abusing antibiotics so seriously that we are now also having antimicrobial resistance to the essence that very soon we may not be having effective antibiotics anymore. These are areas that we need to address very seriously, and it's important for us to look in that direction. Otherwise, very soon, we may find out that productivity will decline so badly, and this will impact on consumption, and also public health will also be negatively impacted. This is very important. And then we have to encourage government and policymakers to look in that direction. Health management in aquaculture is is non-existent. You can't find that at the local government level. You can't find it at the state level. You can't find it at the national level. You can't find it at the regional level because nobody is looking in that direction at all. These things are important. And the world is more or less a global village now. It's in, the global, antimicrobial resistance is a global concern, and we must address that. Thank you very much.
Let me try. Yeah, thank you very much. And then Koficho Kujo. Yes, Kujo, your turn, please. Could you unmute yourself?
Yes, I am doing that now. I just want to say that I'm actually very, very happy the exchange of ideas and views on aquaculture in West Africa or Africa for that matter. But there is something that actually worries me, maybe looking a little bit into the future. In Ghana, the major aquaculture facilities are on the Volta Lake. The Volta Lake is a major source of drinking water. Now the practice of aquaculture, where a lot of nutrients are pumped into drinking water source, antibiotics are used, most likely indiscriminately because there's no pre-regulation. I fear for the future of maybe the water bodies that are being used. And I wonder if farmers and regulators can consider using maybe farming on land rather than in the lake. Because eventually, if the lake is polluted, you destroy a major water body. It was okay initially to encourage the practice of aquaculture on the lake, precisely because that's what we have. We have a shortage of fish. But then as we progress and things are getting more organized, I think one should also start looking at the environmental degradation that this could bring by maybe trying to organize farm on land. There are systems now that can be organized easily. Currently, the pond farmers are doing that. Then you are better in position to also control diseases and things like that, rather than you doing that in the lake. That's my contribution. Thank you.
Yeah. So thank you very much. Yeah. Our next comments or question from Emmanuel Sakite. Emmanuel Sakite.
Hello.
Yeah, we can hear you. Go on.
Okay. It's been an interesting seminar so far. I would like to add up to the current speaker who just spoke about pollution on the lake. I think the solution rather lies in our feed manufacturers lowering the toxins that would affect the water quality. I believe if we culture fish in the lake, we shouldn't have problems in future if the cultural practices are right.
Second floor.
Farming on the land using ponds is more expensive on the other hand. And that will not allow a lot of farmers to come on board as we want to improve employment in Africa. So I would also advise that we take a second look at our feed companies to produce feed that will not pollute the water. I hope that is a bit understandable.
I just left it open. So what? Just descend to the second floor. Okay, but you go there. You take the key with the yeah, that one. Sure. So once you head down. No. Just the second.
Any other contributions, please?
From here. The next floor. No, just descend once.
Right.
Then go through that.
Can I put a question on the floor? It looks like aquaculture in Africa has been limited. The last contributor just talked about us also diversifying into ponds. Like in Ghana, there is so much concentration on production on the lake. Of course, a large majority of farmers are on land already, just that they are small-scale producers. There is room for more production on land. In Nigeria as well, there is a lot of catfish production on land already. But can we also look at the sea? What are the challenges? The researchers here, what have been your research challenges? And the producers, if there are farmers on land, amongst us, what have been the challenges? Why are we not going out to the sea, like mariculture? What have been the challenges? Why are we not practicing mariculture? And the researchers, what are the limitations to your research on mariculture in Africa? Can we just quickly brainstorm that and see how we can improve on that aspect as well? That's a way of diversifying aquaculture in Africa. Yes, Kujo, your hand came up first.
Yes. I reiterate the point I made about fishing or developing huge fish farms on the Volta Lake, precisely because the cost of contamination or pollution of the lake will also not make it possible for you to farm fish in the future either. I think that resourcing small-scale farmers, that may be, okay, I understand that pond construction can be expensive and will pose a hindrance to a lot of the small-scale farmers. But I think food production in Africa will actually have a meaningful impact and generate work for the local people on a small scale than the huge farms. I honestly mean that, because in Norway, where I live, it has taken a lot of money to decontaminate whole rivers, precisely because of contamination from maybe pathogens and so forth. So it is very important that one develop the right practices by either filtering water properly into lakes, so that you can have, you have these closed systems, what you call recirculating aquaculture system, which are closed. And some of them are so advanced in a way that with the right management practices, biosecurity measures, diseases are almost eliminated. Use of antibiotics is completely reduced. Now, when you pump a lot of water feed into a lake over time, where the lake already has a lot of organic matter, what will happen will be a lot of oxygen deficiency in the long run, and the lake will die naturally. That's my concern.
Yeah. So,
Nuruddin, your hand was next. Unmute yourself, please.
Thank you very much. And way forward, I think it is right opposite from what they are experiencing in Ghana. Nigerian water bodies is not even utilized at all. We have just minimal activities going on, and it is recently with advocacy from organizations like FICIN, like TADAN and CAFAN that has it's been a big, huge bottleneck for you to have access to water bodies in Nigeria. So if you are looking at Ghana, I think the speaker is rightly, we should be concerned about his worries, because if all major activities is going on the Lake Volta, there should be advocacy for more on land, so that the lake water, we know what is happening with Lake Chad. I know that one is drying up, but this time around pollution too, might be dangerous. But even though if we are doing land also, some of the runoff water too might end up in the lake. So I think we should look at it holistically in terms of how do we regulate the population or the capacity of fish that is being raised in lake water. And also people encourage more people to do land aquaculture. And when they do the land aquaculture, the feed being used must be looked into, so that more waste will not be put into the water body, and also the runoff. So I think it's a continuous discussion, and I will encourage my brothers in Ghana, the scientists also to look at it holistically and advise the government on the way to go, because we all need to protect our nature. And part of the United Nations Sustainable Development Goal is life underwater too. We need water in as much as we too will live on land. But coming to Nigeria, we will also need more advocacy in Nigeria to enlighten our government, to encourage farmers to go into the water bodies, because we have lot of water bodies that is not well tapped into. So if we really want to meet up with commercialization and producing at a cheaper cost, and also to have this commercial production, we need government to also help us in this space, to create accessible road to the dams, to give licenses to people, to encourage cooperative farming on the dam, and also to provide amenities like storage, where people can produce their fish and store, and also to process, because none of us has ever spoken about marketing, which is one major challenge that we are facing in Nigeria. I don't know about Ghana. I was having a discussion on a platform with a farmer where he posted that he wanted to sell his fish, and he was pricing it that people should come and buy at 700 naira per kilo. And everybody in Nigeria can attest to it that now it is not the same. The price shouldn't be 700 when we're selling 700 in the past 2 years, when we know the cost of feed that has increased by over 50 to 45%. So marketing and processing too, we should look into that, not just producing. If we want farmers to be sustainable, then we should be encouraging them and advising them on how to do processing of what they produce, because market is monopolized in Nigeria. So we really need to come together. If we really want to help West African aquaculture farmers, we need to look at all these things holistically. And that's why I'm very happy, and I feel that such a platform that we have now should be a continuous one. By the time we start doing it, we can take all these messages to the farmers, invite more farmers, and maybe perhaps we invite our government, which I know some of them are already on this conference. Then by the time we keep reiterating it, then the future is bright for the fish farmers. Thank you.
Right. Thank you very much. And then our last hand, Oladosu.
Yeah. Thank you very much. It's been an interesting discussion so far. What I want to contribute is that the culture seems to be a very attractive alternative, except for the fact that it's capital intensive. Apart from that, how many candidates, how many marine species have been developed for culture? Apart from shrimps, which also have very good export potential. Outside that, insecurity, piracy, and the rest of it is a big problem. I think that's affecting those who are using trawlers already, the industrial fishing industry, the industrial fishing sector. They are facing serious problems with piracy and insecurity. Those ones are there. But by and large, Volta Lake will, of course, deteriorate over time if there is constant use and you have increased number of farmers, because fish, the waste from fish will, of course, increase the load year in, year out. So there is no way it will not be necessary to come on land and use ponds. Ponds are far cheaper. And then in Nigeria, for example, a lot of farmers use borehole water to rear their fish in concrete tanks, in fiberglass, in plastic tanks, and they get by. The only thing is that large commercial production may not be feasible doing that, except for flow and
Recirculation
system. And the circulation system also has its own problems. You need 24 hours, is almost impossible. Look at and we brainstorm on this and we refocus our research, we will be able to enhance productivity as we go on. And this is my submission. Thank you.
Right. Thank you. Thank you very much. I think it's been a very wonderful session. The numbers keep fluctuating because network challenges, we can understand this an Africa-wide challenge. And so we have people dropping in and out. We've had up to about 72 participants. That some of the times I check. Maybe those the main host can tell us, but it's gone that much. And then we've had 9 wonderful presentations which have spanned from fish diseases. We've heard of water quality. We've heard about feed and feeding on growth. And then we ended with a presentation on the potential for mariculture in Africa. That has been great. And some of the lessons we have learned is that at least this platform has given an opportunity for collaboration between scientists and farmers and some institutions, government institutions, producers. And so let's keep the network moving. We've also heard people advise that there's a need to diversify the industry. There has been too much focus on aquaculture of catfish and tilapia. And there's the need to diversify the system, the production of tilapia and catfish to other species as well. And of course, also diversify the production system in Ghana, especially from the lake to other systems on land, because the cost of cleanup of the lake will be too high if we wait till we have a dead lake. So thank you very much. The next session starts at 12. So please, we'll beg to end now, and then so that people can quickly freshen up for the next session. It's been a wonderful time, and I'm happy being the facilitator for this session. Thank you, and have a good day.
Do you see potential for economic progress that also improves human and animal nutrition in countries with some of the fastest growing populations in the world? Do you want to partner to find solutions for the protein gaps found in Africa, Asia, and Central America? If you can join us in capturing business opportunities in nutritious foods and feeds, then stay tuned. Visionary U.S. soybean growers founded the World Initiative for Soy in Human Health, or WISH, as a program of the American Soybean Association. We are committed to strengthening agricultural market systems in